Thank you, Arlene. So we've now had a sneak preview at this conference. In addition to everything else that's exciting that's been happening, we've heard a sneak preview of some videos. Phil Brown is not here yet, and two of the sponsors of the conference asked me to welcome you back. Welcome to day two, and for those of you who weren't here yesterday, welcome to this conference. It's been fascinating and energizing. The energy in this room yesterday was practically shaking the windows out, and I expect we'll have more today. And I'd like to, in the interest of time, get us right into the panel this morning. This panel it really follows on something that Sylvia said yesterday in her introductory remarks, which is democratic science, science that connects with people in their communities, and in fact learns from people as part of the science. So that's what this session is to give some examples of. We have four speakers, all of whom have been engaged in one way or another in what I would call democratic science. So they're not going to speak in the order that's in the panel, in the program. They're in the order in which we were originally planning to do this. So I'm going to read you who will speak first. First is going to be Ted Emmett, who's worked in a retired professor from University of Pennsylvania, but who's worked in the Parkersburg, Ohio area, Parkersburg, West Virginia, and I have to look at his Little Hocking Water District in Ohio. The second speaker is going to be Courtney Carignan, who's been working in not only is she working in peace, she lives in Portsmouth, which is, it's a scientist in the community, it turns out, as well as working with the community. So she'll talk about her experience. Third will be Laurel Schaider, who, here she is, has been working in many places, but we'll talk about her work on Cape Cod, especially in the Hyannis area. One of the speakers yesterday described the water system in Hyannis, and Laura's going to talk about some of her work through the Silent Spring Institute. And then the last speaker will be Dr. Frank Bove, who works at ATSDR and who knows all about CAPs. In fact, is deeply involved with the Camp Lejeune CAP, which I think is the premier example of a federal agency doing science with the community. And he'll give examples and reasons why that actually has improved the science. And I think we'll try to do each presentation, if we can, initially, and then have time for Q&A at the end. If there are sort of urgent questions after each presenter, raise your hand, and maybe I'll take one or two, but we need to keep moving so we get through all four speakers. So first is Dr. Ted Emmett.
Thank you very much. It's a great pleasure to speak in an auditorium funded by the proceeds from asbestos. And also it's a bit disappointing, I think, that we always, at the action end, we often get the smallest audiences. And I think that's a sad reflection of some kind. Well, I want to take you back to Little Hocking, Ohio, where I got involved in this. And this is from Little Hocking, looking over from the Ohio River, close to where the water was collected and sent out 25 to 30 miles. And unfortunately, across the river is the Washington works of DuPont that we've heard about. And I want to take you to 2002, because that's when I started to get involved in this and what the situation was. Rob Bellot talked about the safe levels. This place in West Virginia, not Ohio, for the Ohio citizens. West Virginia is setting what they said were the safe levels. Safe levels started out at 1, but it was discovered in the water at 8. The safe level, suddenly they reassessed the information, the safe level is now 15. It was discovered in the water then at 80. The safe level was now 150. Think if you were drinking that water. The Environmental Working Group criticised, quite sensibly, this, I would say, approach. Little Hocking Water decided that it would like to test the water itself. It's sending out the water, and it wanted to know how much PFOA was in it. They weren't allowed. The labs, all the labs that tested PFOA at that time said, no, we won't test your water. We work for the companies. They were unable to test it. Linda, one of the activists on this, is here today. Media reported high levels of concern. 3M says, well, we're not producing this anymore because of, it seemed, developmental issues. EPA declared it was looking at whether this was a probable human carcinogen, and Rob Bellot was initiating his class action suit. And I'll take you, and nothing was happening from government agencies. And I'll take you to a quote next from 2005 from EPA, and that basically said the situation there is that the population source is unknown of PFOA. We know it's in a lot of people all around the world, but it's unclear. We cannot determine how people are exposed. Whether exposures are due to PFOA in the air, water, dust, sediments, dietary sources, or some combination is unknown. We can't regulate that. So we set out and made what we called an environmental justice community partnership. And there were grants for this from NIHS, which we successfully obtained. And that collaboration consists of 3 people. They're not given anymore. They were wonderful grants. An environmental scientist, which was myself, a local healthcare provider. I had a trainee, a former trainee doctor who was a doctor in the area, local health person, and a community association, the Decatur Community Association, people drinking the water. And our objective was to address this information. We set up an advisory committee. It was great. Had all the research partners, of course. Each township in the district was represented. The state EPA, the federal EPA, the county health commissioner, local wonderful person from the school system, the deputy head, a community project, and we met quarterly. Our meetings were open to the public, but we actively discouraged 2 groups, DuPont and the plaintiffs' lawyers, because the community wanted this to be their information. They saw others as having perhaps vested interests in this, rightly or wrongly. And our study objectives were, what are the levels in the people? If there's PFOA in the water, what's in the people? If it's in the people, what is the major source? Is it air, water, other fish from the Ohio River, whatever? And are the blood levels of PFOA, people with high blood levels, do they have more thyroid, liver, or cholesterol problems? And we couldn't look. It was impossible, this first study, to look in detail at developmental effects, cancer and the like. What did we do? Our study methods were to, I have a pointer here. It might take me too long. Maybe you can see this slide better. We took a stratified sample of households. In the end, 196 households, 378 people across the region. And we deliberately had 2 groups. The first, in red, were the people who had a high air exposure. Because we didn't know, is it the air, is it the water? That was a major consideration. And modelling showed us where they were, although there was probably a little bit in the air going out a long way. And then in the yellow up there were other people in the study, and they lived a long way, I think at least 5 or 10 miles over a range of hills to the opposite way the west came, now to about 30 miles. And they had the same water, but did not really have as much in the air. And we thought that'll tell us, that will help answer the air and water question. And we studied people over the age of 2. The 2-year-olds were terrible. Oh, we had to stop. They would cry, and people waiting in the waiting room for the study. It was awful, but it was very important information, as it turned out. And you had to have lived in the water district for 2 years and couldn't have a bleeding disorder because you'd have problems with the thing. And I want to talk particularly about our communication strategy for the results. Now, how we set it up, we had our partnership, we got our grant, we got what's called a certificate of confidentiality, because there's a lot of interest. Oh, my goodness. a lot of interest in this study. And the certificate of confidentiality meant even if we were sued, we didn't have to release the information on people anyway. Very important for this. We selected our study participants. We had a quarterly newsletter. We had a website. And before we had any results, very important, before there's any results, how are we going to communicate them? We're not left with these results. Oh, how the hell do we talk about this? We had to think about this ahead of time. And how we did that, goodness me, this doesn't want to come. The community, we sat there, had a long meeting and determined what did the community want, because this is a community study. And what they wanted is this. They wanted the participants, people put their body on the line, they should get the results first, before anyone else. Secondly, we'd inform the press, timely, and we would try to control the message. It can be done. At least it could be done in this setting. The study must remain credible. This is a community study. This is all our credibility on the line. We must maximise constructive responses and minimise pointless concerns to the findings. And if anyone has a question, it's got to be answered promptly. And here's the order we want the information to flow. The participants, the community, the local EPA and health department, then the nation, then the world and science. That's what we want. And we wanted, as the investigators, some other things. We also wanted, we had informed consent, we had to follow that strictly. Only individuals would get their own result, and only the community, and anyone else would get aggregate results, not able to identify any individual. And actually we had ways for individuals to participate without letting anyone know. We had secret ways they could go to doctors' offices and things. Very few people actually took us up on that. And if you had a medically significant result, one of our physicians would contact you immediately and let you know what happened. And we did have a few. We did have a few. Well, I'll just tell you the results and then go into how we communicate. What we found, the serum levels were very high. 340 parts per billion median, 5 the average across the country. And we had a control group in our study where we took people from Philadelphia, may not be normal, but anyway, their level was 5, just like the rest of the country, 340. Except some people were higher. The children were higher because they take more water in, where the PFAR is, compared with their body mass. They were much higher. The elderly, all the vulnerable groups we wouldn't want, the elderly were higher. More drinks of water you had, you were higher. Adolescents actually had the lower because they drank a lot of Coke and that sort of stuff. Apparently less PFOA. Homegrown vegetables, you were on the average a little bit higher. If you worked at the company, it was basically the amount you got from the company, the amount you got from drinking the water, and they weren't far apart. For the production area, you worked elsewhere in the company, was less. And if you used a water filter, it was lower. Appreciably lower, but nowhere near normal. The source was water. The air, it didn't make any difference. Close far away, it was the water. There were many lines of evidence. I can't go through them all that told us it's the water. And we didn't find evidence of thyroid problems, liver disease, or things. No acute things. And we couldn't be this is an initial study of that, but we did not find those. And we couldn't address developmental defects or cancer in this study. And that the project set up under the lawyers is, really that sort of thing looks at that. How did we get the results out? We had a sequence of timed releases. We had to work this out carefully with the community because the participants are to get it first. Firstly, a participant may be a reporter. So we had to get the results to people in a way that they wouldn't have time to get it to the TV station and the paper. People in the community, they knew all this stuff. we figured it out, and we figured out what post office to go to and when to send them. And we sent a letter to individuals, and we had a little bit of interpretive material with it. I'll talk about that in a minute. Our physicians already, we answered questions. We've got some questions from doctors. We've got some questions from people with conditions. We talked about them. We sent the summary of results, and we had to the authorities, and we had a press release. The press release said basically three things. The levels are very high. The source is the water. Come to the presentation in August the 13th, I think it was, in the high school auditorium, and we'll tell you, and you'll get the rest of the story. And the interpretive, the interpretive thing, we gave them some, really telling them what PFOA levels were, what they were in industry, the highest ever recorded, which was from a plant, Martini plant in Italy, what they were the average down at the plant, which we found from DuPont, what they were in each segment, and the fact that if you worked at both the DuPont, you drank the water was higher. So we gave them this basic interpretive. What's safe? We didn't know what was safe. All we could tell them was what was there in other people. We then had our community meeting. A terrific rehearsal the night before. Everyone swore to secrecy with the and they told us how to present this. What sort of questions will arise from the audience? We spent 3 or 4 hours going over the presentation, modifying, getting all this stuff. It was really, really constructive. And then we had the community meeting. My goodness, the television cameras, the auditorium's full. All these people have arrived in suits. Kelly Lyons in the book described it in suits. God knows where they're from, but they're from out of town, and they're all sitting there, and they didn't know what was coming either. We had newsletter results. The presentation, 45 minutes, 2.5, 3 hours of questions. Wonderful questions. A website presentation, everything went up on the website. And then later we met with EPA and told them what was happening and nationally and our publications and the like. Now, and then later we're able to follow up. I need more than 1 minute, but I don't need too long. What was our recommendation? Consider using some other source of drinking water, and that would be bottled water for all the things, brushing the teeth, drinking, making tea, coffee, soup, etc. What happened as a result of this? First of all, the day of the release, Little Hocking had sent a letter a year and a half ago to asking DuPont for bottled water. Never got any interest. Suddenly, free bottled water. Everyone in the water district. 3 litres a day. Bring in the receipts for the pass, and the trucks will be coming shortly to every street and give them out. EPA followed with a consent agreement on any other community has this, and high levels by today's standard, but any other community more than 0.5 part per billion, they also got free bottled water. New Jersey, Minnesota were able suddenly to release state standards because there was some human basic data that let them make the sense of the animal findings. We did a follow-up 16 months later. 65% of the people participated. What had happened, the PFOA, serum PFOA had fallen on the average 26%. That's about what we'd expect from the half-life. 88% of the participants had gone to bottled water, followed our recommendations. But very interesting, another 7% had changed their water but not done what we said to do. Sometimes they'd put in water filters and other. And as we've analysed this, and we've written about this, what the information produced was what we call a free market of solutions. People didn't necessarily do what we told them to do, or thought they should do. You can't tell people what to do anyway. But they did something that was prudent, something else that they made a judgment with that information and everything. We spend too much time telling people not what to do and not enough giving them information that they can make their own decisions about it. And the community, it was amazing. Little Hocking went from the place over there that drinks wastewater to the little hocking that could. It was an amazing empowerment. And I've got to acknowledge my co-investigators, the wonderful people who worked at Little Hocking, and the NIHS. That fall of grant, the Environmental Justice Grant, stimulated and made this possible. And without this, a lot of the whole story of PFAR, I think, would be a little different. It would certainly be delayed, and especially the public health issue is don't drink any more of that water. That would certainly have been delayed. And there's some references, and I've referred both to some of our papers, and also to Callie Lyons, the chapter on our study in her wonderful book. Well, hey, that's the story. Thank you.
That's a great story. I think instead of asking for questions now, if you can, I'd like to get through 3 other presentations, and then because there's going to be a lot of similar kind of themes, I think, and then we can talk about, questions from the floor after that. With that, Courtney Carignan.
Thank you. Good morning. So as Dr. Clapp mentioned, I live in Portsmouth, New Hampshire. I moved there about a month after the contamination at Pease was discovered. I'd taken a position at Harvard, and my husband taken a position working at Pease, actually. And he came to me and he asked me, should I drink the water? And I said, well, I heard that they took the contaminated well offline. It was probably okay. But, you might want to filter your water just to be safe because, I work in this field. And so I think, that's what he did. And I told him, do you, I kind of looked up, I did a little bit of research, and I said, it looks like there's some associations with testicular cancer. You could tell your colleagues that they could do self-exams. And I'm happy to help if anyone has questions. And my husband's really shy, so I don't know that he even talked to them, but I didn't hear anything about it for another 6 months. And I saw Andrea in the Seacoast Online, and I got in touch with her. And I sort of, tried to put my training to use. So I just want to tell you really quickly what that training is and my experience. I have a BA in biological sciences. I worked for 4 years as an environmental risk assessor. And then part of that work, I did a response action for a community that was exposed to trichloroethylene in their drinking water. And that experience is really what motivated me to get my PhD in environmental health at the Boston University School of Public Health, where I trained in environmental epidemiology and exposure science in community settings. Investigating human exposure to flame retardants, which are different from the AFFF. So flame retardants are a different class of chemicals than PFASs. There's often confusion about that. But I also trained with Dr. Tom Webster, who worked on the C8 Health study. So in our department, I heard a lot about the C8 study and PFASs, so I was familiar with this class of chemicals. I then did a postdoc at Dartmouth in their Children's Center, investigating infant exposure to arsenic. And then I've been training at Harvard in environmental reproductive epidemiology. I guess I didn't learn how to pronounce it. But so anyway, when I learned about this and I got in touch with Andrea, I was really just trying to do my job, trying to put, make that information available to the community so that they could make some, decisions, informed decisions. So my involvement at Pease is I am a Portsmouth resident. Pease is within Portsmouth. It's a trade port. It's on its own water system. There's 3 wells there. 1 was shut down. 2 remained online and now are being treated. And Portsmouth has a separate water system, but there is concern that the contamination may be migrating towards the wells in the what they call the lower well field, the southern well field. Thank you, which is my drinking water. So. I have an interest in protecting my water as well. So I'm on the community assistance panel as a scientific advisor for ATSDR, and I also am on the restoration advisory board as a resident because I live in Portsmouth. So I just want to share with you some quick things that I'm going to clarify about questions that I have in mind and experiences, and then wrap up with the feasibility assessment. So this is a slide actually I got from a colleague in Sweden. So yesterday, Dr. Fletcher talked about Ronneby. I think it was the spring after I started talking with Andrea. I went to the Copenhagen workshop and I met Dr. Jakobsen, and she was talking about Ronneby, and she showed this slide, and I still have not been able to get one for the U.S. I'm not sure if the AFFF used here is different at all from what was used in Sweden. I'm still trying to get that information. If anybody has it, I would love to be able to update this slide. But she had information on the old generation and the new generation AFFF, what was in it. I think what happened in Sweden is really interesting because they found their contamination about the same time as peas. And within a year, they had a health study up and running. They had biomonitoring data. They have a children's study going. And I think we're 3 or more years out. And I mean, we have biomonitoring data, but what's done in Sweden, I think, scientifically has been much more progressive. Often people say, as Andrea said yesterday, that the levels in peas aren't that high, and that's good. People worked there, right? And so they were drinking water when they were at work or when they were at daycare. And so what you end up with is this really wide range. So some people don't have such high exposures. Other people have relatively high exposures. And so this is showing the PFH excess concentrations in children. And I think what's really notable about this is that 40% of the children are above the 95th percentile of NHANES. And so, that's not something that gets pointed out a lot, but I do think it's worth noting and looking into. And then here's looking at the PFH excess levels in the peas adults, which is that yellow line on the top, and then the children, which is the yellow line on the bottom. So these are average and geometric mean concentrations, and then compared to other populations here. Another slide that I really liked that came out of the DHHS report. So this was the biomonitoring that was done on it was over 1,000, almost 2,000 adults at peas, and the over 300 children. Is this when you look at the PFH excess or the PFAS levels by age, you can actually tell us a story. So you start okay here. So you start preschool or you start going to the daycare, and you can see the levels go up in the children as they get older, and then they stop, right? This is around the time you go to kindergarten, and then the levels go down, and then this is about the age people start working, join the workforce, and then you see the levels go up again. It's kind of it's one of the things I love about science. This is I don't know what to say it's beautiful, but it's not beautiful if it's your blood, and you're the one that this has happened to, but it's beautiful in the sense that it tells an elegant story, and I think it's really powerful. So that said, I often get told by other researchers and by government officials that, we're really concerned about stress. Stress is the thing that we can measure in community members when they find out about this contamination, and I believe it. It's very stressful. You go to these meetings and we talk about the health effects, and it's very stressful, especially when you talk about the unknowns, right? I often hear community members say the unknowns are scarier than the knowns. And so I guess what I want to say to that is, I really try to counteract that stress with empowerment, because you tell people, if you try to tell people not to worry and you don't give them the information, then they go and Google it, and they worry, right? And I think what was done in Vermont was really nice, because what we hear from the community there is that, they really felt like their government officials were responding, that they were concerned, that they were being taken seriously, and and that's the type of response that I was trained to give at BU. So that's what I try to do, is empower people with information. I'm not here to make decisions for people. I'm here to try to answer questions and provide information as a scientist. So I often end my presentations with a slide about, health monitoring. What are things that, we get in our physicals that might be useful in terms of, monitoring our health and making sure that we're healthy and we can be proactive about our health. This is something that everyone does, right? But it's something that I think gives people information specific to PFAS exposure that could be helpful. So yesterday I put up some slides about the ATSDR physician fact sheet and about the C8 health study monitoring, and communities tend to really appreciate getting that information. Another thing that's come up is about differential excretion. So we've heard a couple times now that women have lower levels because of menstruation and pregnancy and breastfeeding. I'm really happy there's some benefit to menstruating. I feel really smug about that. But this question comes up a lot about, well, how can I, excrete it more quickly? I really liked Dr. Fletcher's slide yesterday showing the real range in levels in people and talking about that, because that often, really doesn't get talked about. And it's a really interesting question. What is it that makes people have lower levels? It's interesting, I think, from an application, as a community member wanting to know, how can I reduce my levels? Are there interventions? It's also interesting from a scientific perspective, and when we think about misclassification and measuring exposure using biomonitoring. I think that is a very important question. The uniqueness of AFFF exposure, I think, is important. When you compare levels of PFOA in an AFFF exposed population to the general population, you get an answer that is very different than if you look for the range of chemicals that we find in AFFF. And so on the left-hand side, I'm showing the levels that are monitored in Bourne serum as a standard serum panel. And on the right, I'm showing the level, the different PFASs in AFFF, which I got from the Swedish group. So sort of one of the questions that I have is, are we monitoring for all the right chemicals in these communities? Another question, of course, is about mixtures, right? If we're moving from one type of AFFF to another, and this training is still going on, are people, getting exposures to new contaminants? Are we recycling the problem? Same thing with Gen X. We want to get ahead of that, right? So from my experience, actually, I just wanted to mention that I talked with Dr. Brooks, who is the physician who worked at the C8 Health Study and did that amazing recruitment. They recruited almost 70,000 people in 1 year. I didn't realize, actually, until I talked to him about that. And he told me about, the community responded very positively. They did a survey about satisfaction. And the community said that they really liked what he did because they get the results back very quickly. So in research studies, we typically hang on to samples, and then we run them all at once because it reduces the variability and improves the precision, right, of our results. They did this all within a year, which is good. But what they did is they sent the samples off real time, and then they got the data back real time. And so people were getting their results, both their clinical results and their PFAS levels, within, I think it was an average of 10 days by mail, which is amazing. It's sort of unprecedented in my world. But people really liked that, and they would call them up if they had, a clinical result that was abnormal, like a high blood sugar or something that was concerning. And so people were getting health information, whether or not it was related to their PFAS exposure. They didn't know, but it was providing a service to the community. So key components of health study, community engagement. He talked about utilizing technologies to be efficient and effective, doing all questionnaires online, just really streamlining things. I think that's something we need to be moving towards in our field. Being immediately useful, providing when you have somebody participating in a health study, most people don't understand how long research takes. So, coming up front with something that's helpful, and engaging community to find out what is it that you want, what is it that you want to know, what can we answer in the short term, what can we answer in the long term. Tailoring for key outcomes of interest. So you don't, Dr. Webster taught me, you want to make sure that whatever your main question is, you're doing that really well. And then if you can do other things, you want to be flexible. This part I added. But you want to be flexible to unknown outcomes, right? So as researchers, we want to study. We have our main research question, but we also want to be able to answer a lot of other questions. And so we want to make our studies so we can be flexible as well. So I've been working with ATSDR as a CAP scientific advisor. And I just wanted to briefly go over a couple of pros and cons. So this isn't a picture of a CAP because I couldn't get online last night, but this is a picture of just a community meeting. When we do the CAP meetings, we're sort of in a U-shaped table, and all the CAP members in ATSDR sit at the table together, and and we take turns, and we have a moderator. And I think ATSDR has done a really nice job, from my perspective as a researcher that tries to do community-engaged work, in engaging the community and making sure that the community has a seat at the table. So when we first started this process, the people who had a seat at the table were mainly people, that were sort of part of the city in a way. And Andrea was actually like the only real community member on that panel. And it was very stressful for her, talk about stress. And then I remember our first CAP meeting, sitting at that table, and it was a good community. You know, we had Lindsay and Alana and Michelle. And them just echoing over and over again this need for, we want to know how we can protect ourselves and our children now, given what we know, and we want to monitor in the future. And they've echoed that as a group over and over and over again. So I hear you, and I've been echoing it as well. So ATSDR has been really trying, and I'm going to talk about their feasibility assessment in a second. But they also, you know, have limitations, and ATSDR is sort of well known for these limitations. And it becomes, I think, not a scientific issue so much as a policy issue, and how can we empower our governments and our government agencies to do their job and to work for communities. So I'm not a policy person, but I put that to the policy folks in the room. So ATSDR's draft feasibility assessment, I'm just going to review it for you all so you know what's in it. They're taking comments until the end of the month and possibly beyond, but I'm going to try to get all the comments to them we can by the end of the month. They did a review of the literature, and it outlines endpoints that they can study that they've determined are feasible to study at Pease alone. So this is based on the numbers, how many children and adults, may be possible to study at Pease alone, and not feasible to study at Pease alone would require engaging other communities as well. So the proposed child enrollment is 350 exposed and 175 unexposed. So exposed means like drinking the water at Pease, I think before it was, the Haven well was taken offline. So they determined those endpoints feasible to study in Pease children alone include lipids, so cholesterols, glomerular filtration rate, which is a measure of kidney function, insulin-like growth factor 1, a measure of growth hormone deficiency, and overweight obesity. So here are the, I don't know if I have to read them to you, but these are the endpoints that say may be possible to study in Pease children alone. And then not feasible to study in Pease children alone would require engaging other communities just because of the lack of sample size. If you don't have a big enough sample size, and I think Dick Clapp and Greg Howard will talk about this in their workshop, is a health study right for my community? And part of what they're going to talk about in that is the need for a good sample size. If your sample size is not large enough, you bias yourself against being able to reach statistical significance, and so your study is doomed from the beginning. And if you don't reach statistical significance in the past, at least, that has been used as a way to dismiss findings. And in epidemiology, we've been talking about this for a long time now, there's been a real movement away from using statistical significance as a line of whether or not something is good or not, and using confidence intervals and context and considering study size when you interpret results. But anyways, that's still an issue though. In the proposed adult study, they proposed 1,500 exposed and 1,500 unexposed. And these are all the endpoints they say they can study in Pease adults alone with sufficient sample size, assuming enough people enrolled. And then maybe possible to study in Pease adults alone. And not feasible to study in Pease adults alone would require engagement of other communities. And they also have a section on former military service and civilian workers at the Pease Air Force Base, and they concluded it's not feasible. And this is what they wrote, and I just copied it word for word because I did not, sorry. I just want to make it clear, I didn't design the study. This is ATSDR's feasibility assessment. I had not, I gave feedback along the way, as we all did, but I'm really tasked now with giving feedback for this and getting others to give feedback, okay? I think my main feedback for ATSDR is about sensitive developmental windows, and so at least when, in this feasibility assessment and talking about studying Pease, we're 3 years out from this exposure. So in terms of children that actually drank the water, they're actually aging out of sort of being able to look at some of these endpoints that would be useful to look at. And I know that the community and myself have asked our state health department to biobank those samples. It's been repeated. I don't know if a letter was ever written, anything official, but maybe the naivety of thinking you ask your government officials, the community, this is what the community wants. We want you to biobank these samples. They could be useful. And that hasn't happened, and we still don't have confirmation about whether that's been done. And that's disappointing because you have 2,000 blood samples that could be useful. Okay, so sensitive developmental windows. I'm now an environmental and reproductive epidemiologist. I actually study preconception and follow forward. And in my mind, and perhaps this is my bias, but that is a good way to study a population. And so what I'm wondering is if you do a cross-sectional study, can you then do a sub-study that has that aim? And that's all I have. Thank you. And you can email me with any comments about the feasibility assessment. And is it online, Andrea? It's on the Testing for Peace website. Okay. Thank you.
Thanks. I think we're going to just keep moving through this. Laurel Schaider next.
All right, good morning, everyone. My name is Laurel Schaider. I'm a research scientist at Silent Spring Institute, and we're a nonprofit research organization based in Newton, and we have a long-standing connection with the communities on Cape Cod. So I'm going to talk about some of our past and ongoing community engagement and some highlights of our drinking water research as they relate to highly fluorinated chemicals. So we were founded by activists at the Massachusetts Breast Cancer Coalition in the early 1990s. They were alarmed to look at the state's first cancer registry and see that rates of breast cancer were higher on Cape Cod. And they wondered why that was and if there were environmental factors that might be at play. So they tried to figure out what research was going on environmental factors in breast cancer and realized there wasn't much research at all. Most breast cancer research was focused on women who already had breast cancer, on diagnosis and treatment, which are, of course, vitally important as well. But they thought, well, if there are some environmental factors at play, then maybe there's some opportunity for prevention. So some women don't need to get breast cancer in the first place. So we were founded to be they call this a lab of our own, or their own, to be their scientific research organization. And we still have a really special and ongoing partnership with them where we do the scientific research, and they are our activist partners, and they help amplify the results of our findings. So the big thing that we did at the beginning when we were founded was to do a large epidemiological study of women on the Cape. Our study involved over 2,000 women, half who had breast cancer and half who didn't, and tried to figure out what the differences between those two groups were. We learned a few things. People say, have you ever thought about the fact that people in the Cape are older than average? And I assure them that yes, we have taken that into account. So we learned that the higher breast cancer incidence was not just attributable to an older population or use of mammography or in-migration or life history factors. And we learned that women's risk seemed to be higher if they'd lived on the Cape longer. But we didn't really find a single thing or things that seemed to explain the higher risk. We did see some suggestion that women who had lived near cranberry bogs earlier in life, when DDT was used, seemed to have higher risk, and there's evidence that DDT can cause breast cancer. But overall, we really didn't find the smoking gun, and I think that's been a disappointment from the community, and I think they would like us to keep trying to answer that question. And I think one thing that we learned also is that for a disease like breast cancer, it's really important to think about earlier life stages. Courtney just had that example of the sensitive windows of time. For breast cancer, we think that important windows of exposure include in utero, when you're really young, early in life, during puberty, and during pregnancy. And people typically get breast cancer later in life. So it's really hard to go back in time and say, well, what was your mother exposed to when she was pregnant with you? Or what were you exposed to during puberty? And so I guess because we had a hard time like really figuring out what the environmental factors might be on Cape Cod, we kind of switched our approach, I guess, and are taking a forward-looking approach. I mean, we still think epidemiological studies are important, and we try to inform them and make sure they're done well. But our approach now is to say, like, okay, well, what chemicals are we exposed to that may play a role in breast cancer? So, for instance, there are chemicals that cause mammary gland tumors in lab animals. Those seem problematic. Chemicals that mimic estrogen that can feed tumor growth. Chemicals that can change the development of the mammary gland. We heard yesterday that PFOA can alter mammary gland development. We think that can heighten sensitivity. And there's a big push right now. We know that animal studies are a really slow way to evaluate chemical safety. So there's a push right now to develop lab-based studies where we try to mimic what goes on in your body in a cell or in something, basically a very small test tube or a plate in the lab. And we want to make sure those tests are picking up as many of these pathways. It's not just a matter of DNA damage. There's a lot of other pathways we think may be involved. So we identify what chemicals we should be thinking about, and then we also think about what chemicals are we actually exposed to. So we've done testing of products. We've test air and dust in people's homes. We think about things like brominated flame retardants that don't just stay in your couch. They move into dust, they move into indoor air, and can end up in our bodies. We've tested consumer products, and then drinking water is also part of this as well. And we put those together and we say, okay, if this is a chemical that causes basically breast cancer in animals, and it's in your consumer product, or it's in your shampoo you're using on your child, or your couch that your kid's jumping on, like maybe that's enough evidence now to move forward and try to reduce those exposures, rather than waiting for proof down the road that it's harmful, that may never come, and if it does, is years too late, and we've missed an opportunity for prevention. Another thing we learned when we did our research on Cape Cod was that researchers, at least at that time, were reluctant to share their study findings with participants. So when we went into people's homes and measured their dust, we were testing for things that there weren't necessarily standards for. There was no clear benchmark. And so when we thought about how we might tell participants what we found about them, because they said they wanted to know, there weren't a lot of other good examples out there. And scientists at that time had more of an approach of, well, the results are really uncertain, and we don't want to worry people, getting back to Courtney's point about stress. And we didn't accept that response. And so we said, well, if other people haven't developed report back models, then we'll have to develop our own. And so our approach to report back has developed a long way. We used to do print reports. Now we have online reports too. And one thing that people find really helpful is to know how they stack up compared to other people. Maybe we don't have a benchmark or a safe level, but people want to know, am I high compared to other people in the study? And when we test for many different chemicals, they can say, okay, I'm high in these two, and it gets them thinking about, what's in their home, what are they doing that might be making them unique for those chemicals. And our approaches have been followed by many other studies as well. And then we also have been studying water quality on the Cape. So a lot of our work has shifted a little more broadly, but our Cape research has still continued to focus on water quality. Our first executive director wondered if there might be something different about drinking water on the Cape that might be associated with the high breast cancer rates, and are there elevated exposures to endocrine disruptors in the water. So why would we worry about Cape Cod drinking water? We think of the Cape as a quaint and pristine location, but anyone who's tried to drive down there on a Friday afternoon knows it is very busy. And so since the 1950s, the population has grown substantially. Cape Cod is built basically on a big sandbar, and so the soils are sandy. The contaminants can move through relatively rapidly. And the groundwater wells that serve all Cape residents, either public or private wells, are pretty shallow. So there aren't rock layers or necessarily any geological boundaries that separate the pollution sources and the water. Another factor that we think a lot about are septic systems. So I'll talk a little bit about our research on highly fluorinated chemicals, but we also think about flame retardants and hormone disrupting chemicals more broadly. And we tend to think of septic systems as in rural areas or where people are real spread out. But there are pretty dense parts of the Cape, I was surprised to learn, that are served by septic systems. So there's a lot of wastewater going into the ground in a lot of places. On Cape Cod right now, when you talk about water quality, everyone thinks about coastal water quality and nutrients and algal blooms. And people don't really want to talk about drinking water quality. I was grateful for this article last summer in the Boston Globe magazine that talked about Cape Cod's drinking water problem and the challenges that communities are facing. Our executive director, Julie Birdie, was quoted in this and mentioned that we're not just getting one chemical at a time in our drinking water, but really a complex mixture. And we're just starting to appreciate the complexity and the importance of understanding those mixtures. And we think that there can be effects that occur in certain mixtures of chemicals that you don't see if you're exposed to each one separately. So I started working at Silent Spring in 2009, and the first project that I worked on was to do studies of emerging or unregulated contaminants in drinking water. We tested for pharmaceuticals, flame retardants, hormones, a wide range of chemicals. And when we were picking out which chemicals to test for, our director of research, Ruth Anderdell, said, I've been hearing a little bit more about these fluorinated chemicals. Why don't we test for PFOS and PFOA? So I was kind of new to the emerging contaminants game. I'd come from studying heavy metals. So I said, sure, let's include those as well. And it was the first time that anyone had tested for highly fluorinated chemicals on Cape Cod. There weren't conferences on highly fluorinated chemicals at that time. And so we found PFOS and to a lesser degree PFOA, although the detection limit was higher. You can't see my cursor. So we tested PFOS and PFOA in 20 different wells on Cape Cod. And they kind of fell into two groups here. So these are the wells in Hyannis. If you were here this yesterday afternoon, you heard from Mark Els, the town manager of Barnstable, where Hyannis is located. And they had relatively high levels, which we think were related to AFFF from the county fire training area and a local airport. And then there were lower levels down here. I would just point out the scale is on a log scale, so these are orders of magnitude. These lower levels seem to be in areas where we saw pharmaceuticals and other chemicals that might be coming from wastewater. We don't have a ton of information about how PFASs might get into wastewater, but they're in so many products in our homes, it's not surprising that they might be ending up in people's septic systems. We know wastewater treatment plants can have PFASs. So a couple of years later, the UCMR testing was done. I've highlighted in yellow the 2 water supplies in Massachusetts that are on Cape Cod that showed up. So 170 public water supplies tested all throughout Massachusetts. And you can see that Hyannis really does stand out in terms of being having the highest concentrations in any individual sample for 4 of these PFASs. After the guideline for PFOS and PFOA was lowered last year, the Hyannis Water System issued a do not drink advisory for pregnant women, nursing mothers, and infants. They could not turn off enough wells to bring themselves under the guideline, so they issued that advisory while they worked to put in new treatment and secure purchasing from other towns. So again, Hyannis really kind of stands out in terms of having the highest levels. Other sources and other parts of the Cape where these are also showing up. Near Joint Base Cape Cod, there's AFFF contamination as well, and some private wells have been contaminated. There was a big oil tanker truck that's overturned at the Rotary, one of the Rotaries last year, and you don't want that to catch on fire, so they put those firefighting foams on it to make sure that that flammable liquid did not catch on fire. And those are kind of like irregular sources. It's not like something you can look up on a GIS layer necessarily, but sort of anecdotal evidence of other places where these might be found. Before there was a county fire training area, people have asked questions about where did they do fire training before that, and are there sort of old fire stations where this kind of training occurred. We think landfills can be a source, and septic systems, which serve nearly all residents on the Cape and also many businesses. The year after, we did a study of private wells on the Cape. Again, we wanted to include wells with a wide range of likely impact, so some that were in fairly pristine areas and some that were in more built-up areas. Again, nothing close to a fire training area or somewhere that would be a likely source of high levels. And we found 5 PFASs in at least half of the wells tested. I'd stress that the wells we tested were not necessarily representative of all the wells on the Cape. We did oversample ones that we thought would be at the higher range of impact. Nevertheless, we did find 5 showing up frequently, both some of the long chains, PFHXS and PFOS, and some of the shorter chain replacement compounds. And in many cases, we think septic systems are the main source. This is just showing if we sort the wells by nitrate levels, The most common source of nitrate in Cape groundwater are septic systems. So if you move over to the right, those higher levels of nitrate are telling you there's probably more septic system impact, and we see higher levels of PFASs. So this is the results for 18 of our 20 wells, and then there were 2 that just seemed unusually high, we looked at the maps and tried to figure out where these might be coming from. One of these might be impacted by a landfill. Another one is kind of down gradient of a commercial area, so it's possible. And these are used in floor waxes, or if there are businesses that are maybe washing carpets, or I don't know. There's a lot of possibilities for other wastewater sources that aren't well characterized. So in terms of our community engagement, we've had a long-standing relationship with people on the Cape. We have an outreach coordinator, Cheryl Osimo, who lives on the Cape and is on, our trusted source of information and really our key link to the community. And we reach out to the community in different ways. So one is by writing editorials for the local Cape Cod Times. This was one that we wrote last fall, trying to urge a more precautionary approach to water protection. The wells in Hyannis where the high levels of PFASs were detected are not in a pristine area. We know in the past that they have VOCs from the airport and perchlorate from the fire training area. So even though for a long time everything was below a standard or a guideline, it still didn't seem like the best idea to be pulling from this area. And so how can we promote maybe pulling water from a different area and taking steps when it isn't a crisis, when you're not over a guideline, in sort of a longer-term planning way? I think a lot of scientists kind of shy away from talking to the media. I know we've heard a lot yesterday about the importance of journalists, but scientists are really concerned about being accurate, and I think we've all had this experience of talking to media and then reading the article and being like, oh, that's not very accurate. We're really cautious about how our work is conveyed. But we also see the media as an opportunity to reach a broader audience. We can have a community meeting and we get the same people that come every year. Maybe we get a few new people, but we want to reach people who don't necessarily even know about us. So we do write press releases when we release new papers, and both on the Cape and just sort of our broader outreach strategy. We had a meeting last month, actually, with Phil Brown and Sylvia from Toxics Action Center, and Courtney was Skyping in from Portsmouth and testing for PEEPs. Alana Davis was also Skyping in. And we let people know about a study that we're planning to do on the Cape. We have a fall community update every fall to let people know about our current research. So I'm pretty much done here. I just wanted to think about what kinds of questions I get. I'm kind of the water person for Silent Spring. So people say, is my water safe? And that can be a hard question to answer because, I've focused a lot on Hyannis, and I don't want to give you the impression that everywhere on Cape Cod the water is really bad, because some places the water is quite good. By contrast, say Brewster pulls their public water from under a state park and from some conservation land, so they actually have really good water quality. So there isn't one answer for all of the Cape. People ask about water filtration. People say, did my water make me sick or my neighbors sick? And then it's hard. Sometimes people say, well, I've had breast cancer and I have a few friends who have. Maybe you could study us. And I want to be really sympathetic, and I appreciate them wanting to learn something from what they've been going through. But we kind of did that. We had 2,000 women in our study, and I don't know. For us, it wasn't I think the way to move forward to help and provide new information. But it's hard because people have been touched so personally by this. So with that, I love you to go check out our website, silentspring.org/water. We post all of when we write a research study, if you're interested in really getting into the nitty-gritties, we pay for the open access so that everyone can read our articles. We write press releases and fact sheets and tip sheets to provide different levels of communication for our study findings. So, thanks. Frank doesn't have slides, right? Okay. So.
The letter D, Thanks, Laurel. I've never heard her describe herself as the water person, but I can see why. Also, I want to say a little bit about Cheryl Osimo, who Laurel and Cheryl wrote a op-ed, I guess it was, for the Cape Cod Times. Cheryl is an amazing person. She's a Cape Cod resident. She had breast cancer. She would not call herself a breast cancer survivor. She calls herself a breast cancer victor. And she's determined to, as Laurel said, be the strong liaison to the community. So we're going to get ready for the Q&A, but first I want to introduce a little bit more about Frank Bove. You can read his current position in the bios for this conference, but you should know that Frank was a community organizer first, and he worked in the Boston area as community organizer, and he and I were both in something called Science for the People. So he's not the typical federal employee, and as you'll see. And I think what he would probably say, or I'll say it for him, is that now he's doing science with the people. And this has progressed. So take it away, Frank.
Good morning, everyone. Since I'm supposed to be representing ATSDR at this conference, I thought I would go over how you can petition my agency for an investigation, and also talk briefly about how CAPs are set up and how often we do it and so on. So first, how to petition my agency for an investigation. Well, it's pretty simple. First, you try to collect as much information as you can on the environmental situation, any health information you get, any number of people who have cancer or whatever in your neighborhood or whatever, and you pull that information together. And I always say it's important to get a supporting letter from an elected official. That always has more weight than if you just send a letter to ATSDR. But that's basically what you do. You collect that information, you send it to ATSDR. You could send it to Pat Bricey, who's the director of my agency. You can send it to Ileana Arias, who's the director of the Division of Community Health Investigations, which actually receives these petitions and looks them over and decides what to do with them. ATSDR does some preliminary data collection of its own, or data investigation, to see whether it's what we could do with this for this petitioner, or whether we can't do anything. Sometimes we ask the state to respond to the petitioner. Sometimes we do it ourselves. It's always helpful also, we have regional offices, the same places where EPA has a regional office. So it'd be good also to talk to the regional person in your area if you want to petition ATSDR to do a health assessment or some kind of investigation. But what I always tell people is that, if ATSDR decides that it wants to do an investigation, make sure you insist that some kind of community participatory mechanism gets set up, that you're at the ground floor of whatever that investigation is, so you can jointly determine with ATSDR what will be investigated and how, and you watchdog the process from beginning to end. So that's how you do a petition. I'm sure that's not the way my agency necessarily would put it, but that's how I think you should go forward with that. Now, CAPs. We set up. We have a Federal Register notice in 1992 to set up CAPs, and we wanted to avoid the FACA laws, so we call them a community assistance panel instead of a community advisory panel, just to avoid all the FACA requirements, make it more flexible than the FACA laws require. So it's a non-statutory group, provides a mechanism to exchange information with the affected community and ATSDR. And it has a couple of objectives that are pretty much similar to all the CAPs we've developed, and including the Peace CAP, and that's to facilitate communication between ATSDR, state officials, and the community, convey community health and environmental concerns from the community to ATSDR, and find out what the health concerns are and other environmental concerns of the community. So in other words, a two-way conversation between the community and ATSDR. Now we set up a few CAPs in the 1990s. What comes to mind is Kelly Air Force Base, Otis Air Force Base, which is now called the Joint Base or something like that. Maybe one or two others in the 1990s. And none of these CAPs really worked that well. I mean, they worked, they had their pluses and minuses. But by the end of the 1990s, I don't remember any CAPs at all that the agency was setting up. So it's not something we do routinely, okay? And around 2003, I started getting involved with the Camp Lejeune project in earnest, and thought that a CAP was necessary. Camp Lejeune is different from most sites because people like Wordsmith are dispersed all over the country, right? So it's a virtual community, if you will, as someone said yesterday. But I still thought that a CAP would be crucial. We were already having difficulty communicating with the community, the affected community. There was a lot of rancor, a lot of distrust of ATSDR for good reasons. We have a history. And So I was trying to get a CAP formed, but I was getting nowhere within the agency. So at the same time, there are two organizations at Camp Lejeune of activists. They didn't necessarily agree with each other either, but they both had websites. And through one of the groups was able to convince Congressman Congressman Dingell back then to push ATSDR to look into adult health issues. We were studying children. We had looking at birth outcomes, looking at childhood cancers. We had those studies started, but we had no plans to do an adult study at the time. So they were insisting on it. So through that intervention with Congress told us to set up a science panel, which we did. And two of the co-chairs of that panel were old friends of mine. And I said, look, if you're going to recommend anything, recommend that a CAP be formed, because I can't get anywhere inside the agency. So this is a strategy I've used a lot, is to get the community people to push from the outside while I push from the inside. It works a lot of the times. Anyway, so the science panel recommended a CAP at Camp Lejeune. I said, great, and I wrote up what we should do, and a CAP was formed. Now, how do we set up the CAP at Camp Lejeune? Was we asked the two organizations that were already in existence. We said, nominate 4 people. And so that was the first CAP. And then we asked them to pick 2 technical advisors. I was pushing for Dick Clapp, and he graciously joined, and for many years was on the CAP as a technical advisor. And so that's how the CAP formed. And it was formed in 2006. It's still meeting regularly. We have monthly phone calls. We meet now 3 times a year, including one meeting somewhere in the community. We've had meetings in North Carolina. We've had a meeting in Florida. I think the next meeting is in Pittsburgh, I think, or we're thinking about that. So we're trying to have meetings in other parts of the country because, again, this is a virtual community. The Peace CAP is a community, so that's a different situation. So there, when we first got involved with Peace, and we came up here in October of 2015, I think it was, we went over what we could do, and at that meeting we were talking about setting up a CAP. Now there was a community advisory board that was mentioned by Courtney that was made up mostly of public officials, and Andrea was on it. And I guess that worked to some extent, but we wanted to set up a CAP which was community of people, not just public officials. Some of our CAPs in the past had a lot of public officials on it, and we have a few on this one too. So we got the information out to the media. We talked to Testing for Peace to also nominate people. We got a lot of nominations, and then ATSDR selected people for the CAP, and we have about 13 or 12. I can't remember how many we have. A lot of people on this CAP. So that's how that was set up. So we had no CAPs until the Camp Lejeune CAP in 2000, since the beginning of the century, and now we have a Peace CAP. So we have 2 CAPs at ATSDR. That's all. So it's not something we do routinely. I think we should. I think we should have some kind of participatory mechanism set up no matter what investigation we do, whether it's a study, whether it's a health assessment, whatever. It doesn't have to be a CAP. I've done a cluster investigation in Brick Township, New Jersey, where it was just a communication between me and the family that identified the cluster, and that worked well. So it really depends on what works in the community, what kind of participatory mechanism will work. So a CAP is sometimes useful. There are other mechanisms. So I was going to tell you a little bit about my background, but Dick already said it. So, but 2 things I think as a scientist. I mean, I came to science as an activist and got interested in epidemiology because of my activism. Actually, involved in both energy issues, environmental issues, and occupational health issues as an activist first. So I took no science courses in college, for example. So I had to teach myself, and that's what a lot of activists do. They teach themselves. That's true at Peace. That's definitely true at Camp Lejeune, and I'll talk about that more. But anyway, so there are 2, the precautionary principles brought up a lot, and there are 2 key points of the precautionary principle that don't normally get emphasized, and I want to emphasize them. First off, the scientists who work with and for communities, as Dick was saying, should be transparent in their objectives, their methods, including how they're going to interpret the data, how they're going to assess causality. All the methods need to be discussed with the community at the ground floor, and clear rationales put forward, and listen to the community for alternatives. Oftentimes the community will have an alternative approach that's better than yours. That happened in my experience with this autism cluster at Brick Township. The community actually had a better approach than I was advocating, and so we adopted that. So that's, the community can improve the science. It's something my agency has a hard time understanding. And I mean, when I say that my agency now, I'm talking about in CDC as a whole. Secondly, that's one thing that's part of the precautionary principle, is being transparent. The second one is involving the community at the ground floor. You don't usually hear that with the precautionary principle. That's a key element of it. And in particular, setting up some kind of, as I said, community participatory mechanism that makes sense for the community, so that the community has the opportunity to challenge the assumptions you're making, the methods you're proposing, and so on. And we have that dialogue. And also, by doing that, you facilitate the development of what we call citizen scientists, so that you're empowering community people to and helping them develop their own expertise in the science, so that they can carry on the struggle in their communities when you're gone, because they're going to have to deal with the environmental threats in their community, and getting them to that point where they can actually do the research themselves is important. It also demystifies science, and I think that's helpful. And also increases science literacy and makes people more appreciative of science in a time when science is under attack, and we've had marches for science. It's very important that the community people see that the science is done for them, that the interest of the science is for the people. Okay, so let me talk about Camp Lejeune real quick. And the Camp Lejeune cap has been extremely effective. Although there's been some turmoil in the cap over time, there are a couple of people in particular that work 24/7 on this issue, that reviewed tens of thousands of documents, that have educated themselves on water modeling issues, on the causes of the carcinogens, and the health effects of other health effects of these chemicals, and so on. And we couldn't have done the studies we've done at Camp Lejeune without their help. There were times when we had to deal with the Department of Defense in terms of funding, and they were instrumental in making sure that we maintained our funding so we could do the studies, we could do the water modeling that was done there, which was pretty cutting edge. And they're still helpful. When we had a difficulty recently with the DOD in terms of funding this new study we're doing on cancer incidence, they got their senators to write a letter, and we have our funding. So so they've been very effective. And one of the ways they've been effective is they've really done the research. And as I said, they've gone through tens of thousands of documents, maybe more, many tens of thousands at this point. They've compiled this information. They've gone and talked to their senators and Congress people, well prepared, with also with explicit things that the senators can do to help the cause. So that's the first lesson: is if you're going to go fight for funding or whatever, and you go visit your senators and Congress people, make sure you're extremely well prepared when you do so. It's always helpful also to bring numbers with you. I think that's always a good lobbying technique. They built this whole cap is an alliance between retired Marines, their dependents, their spouses, and civilian workers at the base. So that's another thing. This was discussed yesterday a little bit. For example, at Wordsmith, the key thing there would be if the community would find ways to build alliances with those retired Air Force people, the retired civilian workers, and because you have a common interest here, you've all been exposed, and see if that couldn't, with that combined alliance, push for funding or whatever you want to push for at Wordsmith. So that's the first. So the second thing the CAP was able to do was had to do was confront institutions. The first they had to confront the Department of Defense, the Marine Corps. Marine Corps was not very cooperative. Marine Corps held the information about the contamination at the base for many years without notifying the people. Actually, the first time that a lot of them heard about it was when we issued a health assessment at Camp Lejeune, which was terribly flawed. So they were able to confront both the DoD, then they had to confront the VA, sometimes they had to confront my agency and CDC, and even the National Research Council and National Academy of Sciences. So there are a lot of obstacles in their way, and they did their job, did their research, organized, and fought them all. And we were very successful. And in fact, they've been able to get something that's never been done, I think. They've been able to get presumption from the VA for certain diseases so that you don't have to prove that your exposure caused the disease. They presume that it did. All you have to do is show that you were there at the base at a certain time. So that was a crucial victory. Another victory was getting President Obama to sign legislation for health care for 15 conditions for families as well as the Marines, retired Marines. So those are two huge victories that they were able to accomplish. But the other thing is that we couldn't have done the science without them. I mean, they had local knowledge that was crucial for the science. For example, we didn't know where barracks were located on base back then. The Marine Corps wouldn't or couldn't tell us. And so we had to ask the retired Marines where they thought the barracks were. This is important because this is one of the ways we defined exposure. They told us about the activities, the training on the base, so we had a good sense of where people consumed most of their drinking water. It wasn't at home, it was in the training in the field, for example. They knew about the wells that were used. We got misinformation sometimes from the Marine Corps about the well uses. Maybe they didn't know, maybe they, whatever. But but this is a problem working with the DoD, and I can talk a little more about that later. But they knew about the well usage. They knew about the extent of the contamination. All that information was crucial for our studies. We could not have done the studies without that important information in order to assess exposure. So they improved the science. Okay, they made the science possible, both from the funding end, but even in the content of the exposure assessment. So I think my time's getting close to being up, so I think I'll stop there, and I'll be open for questions later. There is a documentary about Camp Lejeune it was released in 2011, so it's outdated a little bit, but it's called Semper Fi, Always Faithful. It's very powerful, so if you have a chance, look at that, and you'll get a sense of what the CAP was like up until that period, and I think it's informative. There's also a book that's also very outdated that came out in 2014 called Trust Betrayed. And so if you want to know more about the Camp Lejeune story, that's good places to start.
So we're running a little late. I feel like the sports announcer that says I'm a little pressed for time, so here are the scores: 3-2, 5-1, and 2-0. There are mics around. Questions? Yep, start here.
Just wondered, the overview of feasibility assessment epidemiological study this year said that there wasn't, here's that, you said it wasn't feasible to do epidemiological study on that. Okay. That case, and what would be the justification for that?
So I wrote the feasibility assessment. What we said, or what I said in it, was that you can't just study P's, okay? You won't get enough numbers. So you'd have to include other bases, like Warminster, Willow Grove, Wordsmith, so on. But one of the problems with all these bases is that they had trichloroethylene contamination, especially Wordsmith, but also P's, and not as bad as Camp Lejeune, but bad enough. And so you have to take that into account. So that adds complexity to it. But no, the reason we say that is just that you can't just use P's. It's not enough population. We need more bases involved.
I'm all for that.
Yeah.
And I'll talk to you about the
I have a couple of quick questions. One for you. The ATSCR petition, the ATSCR, I'm sorry, not for you, for the last week. Is that available for any community or only federally designated Superfund sites? So the site I'm working with is a New York State designated Superfund site, but not federal. And could we petition ATSCR in that case?
What, to get involved or
Yeah,
to set up a cap?
as you described that petitioning process, is that only available to communities that are federally?
That's any.
Anything, okay, thank you. And then on the biobanking that was recommended, again, we have a couple thousand blood samples that have been taken from Newburgh now. I'm looking around for this. Oh, there you are. Sorry. So we should be asking our state health department to save some of those samples or save a portion of them. Can you just give me any insights into how to make that request specific enough to maybe be successful?
I think maybe make it very public.
Yes.
And I was talking to somebody last night who recommended actually doing campaigns to, and this isn't something that I do, but she said that what communities can do to get the attention of their health departments is to do campaigns where you're calling and emailing and writing letters, making it very public and very official, that you're making this request. And Andrew, I think you have more insight about why that request has been ignored. It's one thing that they say is that they need consent, and it wasn't consented. They didn't put it in the consent agreement to biobank the samples, and repeatedly the health department has said that it's not a research study. I was talking with somebody last night who said, why do biomonitoring if you're not going to learn more from it? We do a lot of looking at biomarkers, so we can look at biomarkers of exposure, like when we measure the PFASs. We can look at biomarkers of effect, which is measurement of a lot of the different outcomes that we've talked about today are actually measures of effect. And we've gotten more sophisticated, and we'll probably continue to get more sophisticated in our ability to do that. And so, even if you can't think of anything right now, which I can think of lots of things, what we might be able to do in another 5-10 years to see that it seems worthwhile to have them.
In the back, Alan?
Just a brief follow-up on the biobanking, because we've done that. We have about 66,000 specimens, here to take a few hundred. From PFAS activity, it's really good if you're thinking about biobanking. Think about it before you draw the blood. If you do it afterward, there are so many barriers to doing it well that it becomes extremely difficult. Things you have to think about: chain of custody, in addition to permission. It's really good if you have it be up front. It's not maybe necessary. It really helps people if you do. There's a lot of things.
Somebody has a microphone.
Okay, I'm not sure I can be heard. I'll repeat fast. IRB, informed consent. IRB may not be necessary, but good to have. Informed consent is almost necessary. If you don't have it, I think it's a very big barrier. And chain of custody and conditions of storage, that's not a complete list. Biobanking is becoming, the barriers to biobanking are becoming larger. It's not necessarily good, but it's a fact. So think it out in advance and talk to your tissue banker before you draw your bloods.
And I would add to that robotic freezers. Finding samples and shipping them is a huge obstacle, and they actually have robotic freezers now. We put a barcode on the bottom of all the vials, and do you guys have that?
Yeah, with barcode.
It's amazing.
Yeah.
Yeah, robotic freezers.
Usually when you draw the blood, you do it with If you draw the blood in the lab in mind, they would usually barcode. And then any tissue bank that you go to would usually be able to create a dummy barcode so that the barcodes match and you have privacy. So as long as they're barcoded to begin with by the lab you're using, you can probably figure that piece out. So, but if you draw the blood and you're not barcoded, you're right. I mean, that would be extremely unusual. It would mean you didn't have a lab in mind, but you do need a barcode.
Well, with the robotic freezers, they put the barcode on the bottom. So if you do it traditionally, it's not easy to switch over to robotic freezers. You have to sort of do it up front. It's expensive.
It's actually not hard.
I'm going to exercise the chair's prerogative here.
Sorry.
We could have a separate session.
IRB.
Yeah, IRB just stands for Institutional Review Board, especially
Oh,
for
okay.
university-based researchers. You have to basically have that protection, that you've had training in how to maintain confidentiality, privacy, and that you're going to store information in a way that doesn't divulge people's individual results or individual medical histories, for example. And now Claire is giving the sign, time is up, because we need to be able to have at least a 10-minute break. You can obviously continue the conversations, and like I say, the energy in this room will explode the windows. Keep it up.